Um, Hello, everyone. My name is Louis. I am currently a researcher at Vogelwarte here in Switzerland, but this talk will be about my PhD, which I did several years ago now when I was at the University of Science and Technology in Trondheim in Norway. And I did my PhD as part of a big European Horizon 2020 project called African Bioservices. And I studied how impala, a common antelope in Tanzania, adjusted or responded to stressful environments in the Serengeti. So this will be my talk about that, and obviously it will not be about birds, and I have very, very few bird examples, but hopefully I can toss one or two in there. Yes. So first I will give a very soft introduction to disturbances and disturbance risk hypothesis. And then I will set the stage for my PhD, talking a little bit about animal physiology and stress physiology and how we measure it and use it. And then the last part will be about what I found in my PhD. So first, about disturbances to animals. So obviously there's a lot of different types of disturbances that animals deal with, but a major part of that has become human disturbances, both lethal and non-lethal disturbances. These non-lethal disturbances can obviously have many different forms, such as light and noise pollution, tourism, traffic, and these can or have been shown to affect animal fitness through adjusting or affecting their reproductive behavior, affecting population dynamics, or interfering with community interactions, and many more. So human disturbance is often perceived by animals as predation risk, and as such, they will often respond to that by anti-predator behavior. For example, increasing vigilance, changing flight behavior, adjusting their activity patterns and movement patterns. And this is basically what the risk disturbance hypothesis is, that when an animal perceives a certain risk, for example, human presence or disturbance, it suffers an opportunity cost. It will spend energy on fleeing from humans instead of spending that energy on fitness enhancing activities like mating or foraging, and that has then downstream fitness consequences. And so this is basically where my PhD looking at these non-lethal human disturbances as an effect on populations, impala populations. Yes. Quite recently, I went to a conference in South Africa, and two professors, Michael Clinchy and Liana Zanet, were presenting this really quite great talk, I thought, about the human super predator. They've done a lot of research around the globe with lots of different animals, figuring out exactly how much of an effect human presence can have on animal populations. And they've also published tons of cool papers like the ones listed here. But the talk that was present on that conference, you can find on this shortened link that I put here on the bottom. It is really a fantastic talk, and this talks about like exactly that humans are perceived as sort of a super predator, having tremendous effects, non-lethally and obviously lethally as well, on all like all kinds of groups of animals. So one of the major ways that we've been studying these non-lethal effects, or the responses of animals to these non-lethal effects, are through direct observation by looking at vigilance and flight initiation distance. This is also what I did in my PhD, but I will not talk about this today. And of course, using technological advances such as GPS tracking and accelerometers and wildlife cameras. Also something another aspect that we applied in this PhD, but also something I won't talk about today. So today I will talk mostly about the physiological toolbox, or the physiological stress response. So animals can respond to the human disturbances through two main routes, behaviorally and physiologically. And the physiological toolbox, or physiological stress response in this particular case, is quite interesting and offers a lot of opportunities because of the mechanistic understanding that it often allows us to have. So knowing how an animal responds physiologically often gives us the direct cause and effect relationship between threat and consequence. And that is quite important for conservation managers. And what I always liked about this, and what I often said in presentations, was that conservation physiology can often be used to see or discover potent negative effects on populations without seeing the actual population decline yet. Like, you could try and see if something is wrong with the population or something is affecting a population without the population itself going down, which is what traditionally is often the main indicator of a population in distress. So conservation physiology is a relatively new field, although the first research in this kind of category was in the 60s and 70s. Wikelski and Cook started a journal called Conservation Physiology in 2006, and they published the first paper in there, which basically summarized all kinds of tools that were already being used. And they range from studying the nutrition, metabolism, immunocompetence, and so forth in all kinds of animals in all kinds of systems. And this has really taken off since then. This physiological response to disturbances can be measured in many different sample media. Feathers, for example, blood, tissue, saliva, urine, fur, baleen in baleen whales, and even respiratory vapor, which Kathleen Hunt has kind of pushed or made possible in the last decade, where you kind of fly either a drone over a whale spewing out vapor, or you literally hold a long stick out, but then you have to be really close to the whales. But yeah, so there's tons of different sample media you can use, and they vary from being more lethal to non-invasive. So obviously hunting would be the most lethal data collection that we can do, but I would say that internal biologgers or collars or blood collection is also quite invasive, while passive hair collection or fecal collection is very non-invasive, or can be non-invasive. And that is what we did. We used feces because of its non-invasive nature and because it really does offer a wealth of information. Obviously, you can get genetics from the individual that produced the fecal sample, but you can also get information on its diet, the genetics of the plants or animals it ate, hormone metabolites, immunocompetence, stable isotope, microbiome, and so forth. It really is like a kind of a microcosmos of information in there. Yeah, but so we focused on the physiological stress response, which basically means that we looked at, or we measured the concentration of glucocorticoid hormones. In birds, that is often referred to, or the most common there is corticosterone. In mammals, it is cortisol, but both are glucocorticoids. And from experimental research and biomed research, initially we came up with researchers came up with the hypothesis of the cord fitness hypothesis, which basically states that with an increasing baseline glucocorticoid level, your fitness would decrease. And this came to be because of when we experimentally increased glucocorticoid levels in all kinds of animals, we saw that they had negative effects on their digestion. They would have reduced growth, reduced reproduction, immunocompetence, and increased oxidative stress, and many more pathological effects. Sapolsky 2000 was like the main paper introducing all of these pathological effects of chronic stress. So long-term increased stress. And stress in this sense would be an increased, a sustained increase in glucocorticoid hormone in the blood. Now, since then, we've come a long way, and we've gone away from this whole cord fitness hypothesis and saying that stress, whatever that means, an increase in glucocorticoids is bad. Quite the contrary, stress is an adaptive response, and an increase in glucocorticoids might mean a very positive thing. And a decrease in glucocorticoids might actually mean that the glucocorticoid response or the stress response is compromised in animals and might actually be more pathological than an increase in glucocorticoids. A sustained increase in glucocorticoids might also not necessarily mean that an animal is in distress. It really depends on many factors. And so Michael Romeo and Rudy Boonstra, especially Rudy Boonstra, has written several papers on this, kind of going back to the basics, back to the evolutionary origin of stress and the adaptiveness of it, and then trying to like interpret results that we see in these quite traditional papers more in that light. So I think these papers are fantastic and definitely worth a read. And then I wanted to mention one last thing, something that people have been working on for the past, I would say, decade, but it's still not all the way there. So one of the very cool things that glucocorticoids or any kind of hormone does in tissue is that it gets deposited there, whether that's through the metabolites of those hormones or hormones directly get deposited in tissues. And if you have very long growing tissues, such as long hair, big feathers, or baleen in whales, you could show a sort of timeline of stress or timeline of physiological state of an animal, which has been shown by Kathleen Hunt in whales, looking basically taking a big piece of baleen, cutting it in small pieces, and then tracking back what the physiological state was, say, a year or 10 years ago, depending on how long the baleen grew. You can do the same in birds and in mammals with long hair, but there are many pitfalls while doing this, and the methods are definitely not fine-tuned properly. Yeah, there's a good paper here produced by Vogelwarte, kind of giving a sort of literature review on this and yeah, indicating where the next steps could be. But it is a very interesting topic or opportunity, I would say. Okay, so in summary, so non-lethal disturbances can affect fitness, and animals respond to these non-lethal disturbances both behaviorally and physiologically. This physiological stress response offers this mechanistic understanding of how threats affect or potential threats affect populations, which can be very interesting for managers. And there's a possibility of non-invasive sampling, which means that you can get a lot of information on an animal without actually affecting it in any way, or almost in any way. When I say, so non-invasive means different things for different people. To me, non-invasive means that I don't have to touch the animal to get the sample, but I did observe the animal and I was in its environment. So I guess I was disturbing it slightly just by being there. Okay, so now for my PhD thesis. So I worked on impala in the Serengeti ecosystem. And what I basically did in my PhD was to try and figure out how human disturbances, climate perturbations, and social environment affects both the behavior and the physiology of impala. I looked at both thyroid hormones, which I talk about slightly or shortly in a bit, and glucocorticoids. And I took fecal samples to then analyze them. And a little bit background about the study animal and the study area. So impala are beautiful animals. They're sexually dimorphic. They're medium-sized, so they're like a roe deer-sized animal, and they are very common in eastern and southern savannas, or eastern and southern African savannas. They're sedentary, so they have very small home ranges and have very limited movement, both within and between seasons. They live in groups. So there's two major groups. So there's a female herd, which is a big group of females and young individuals that often has one male or adult male with them, and then a bachelor group, which is just a whole group of young males. And then sometimes we can see solitary males. the groups can vary from 5 individuals to 120 individuals, and impala are territorial, so they keep territories for a short amount of time, and then they get defeated and they get replaced. These territories are held in or seasonally, but this becomes less obvious in the Serengeti, which is very close to the equator. So why do we study impala? Well, first of all, because it's widespread and locally abundant, and this is important when we want to collect a lot of samples. They have limited movement, which means that whichever physiological state they have can be linked to the environment they're currently in, because they didn't really move through this landscape very shortly. They didn't get affected by different landscapes or different environments in the past. Basically, they just stay for a long time in the same area. And it's one of the last ungulates that inhabits the human-dominated areas of the system throughout the year, which is a bit unfortunate, but yeah. So the Serengeti ecosystem is located in northern Tanzania, close to the equator, and is therefore quite seasonal with its dry and wet seasons. So no summer and winter season, but there's rainy seasons and dry seasons. And of course, it's a very famous biodiversity hotspot. Yes, for conservation biologists, I think it is a unique and super interesting system, because it has this national park, the Serengeti National Park, in its center. And then it has 6 buffer areas around it, which have all kinds of different regulations, and they allow different forms of human activity, kind of allowing us to do sort of natural experiment there. And so the main human disturbances outside the park are livestock herding and agriculture, and inside the park, obviously, it's tourism. And there's very little tourism outside the park except for Ngorongoro, which is here. Yes. So it's the human disturbances and the social context are quite straightforward, but the climate variables, or the climate proxies that I used, I wanted to just delve into a little bit. Oops, there you go, because I thought they were quite interesting. So we used remotely sensed data only for environmental disturbances. So we looked at foraging quality, temperature, and rainfall. And for foraging quality, for example, we used NDVI, which is a very, very common proxy to use for sort of primary productivity or greenness of a landscape. Basically, the greener this area looks, the more green the grass is. And with this very big seasonality, you can see here, this is a loop for 1 year in 2012. You can see the dry season drying up the entire area before the rains come back and then make it green again. So why did we use remote sensing data? Because field measurements of these things is quite difficult. There's only one weather station in the Serengeti National Park, and it's not very trustworthy. And these data are available back in time and for a large spatial and temporal scale. So this allows us to extrapolate and to link local environments to specific samples that we collected, which would not be possible in any other way, really. Yeah, and so and then a little bit about sample collection. So I collected about 600 and a bit samples over 4 years in both a wet and dry season. And the reason why I mentioned this is that we collected data only, or samples only, from individuals that we knew the sex, the age, and the body condition of. And this is important because physiology, although it's very interesting and allows us to do a lot of things, there's also so many pitfalls. And one of them is that the individual variation is huge. So just age and sex and pregnancy status, for example, body condition can affect the glucocorticoid concentrations dramatically and basically mask any pattern that you would want to observe. Now, we could not observe pregnancy status. We tried to analyze estrogens and progesterones, but we ran out of money. So we kept it with thyroid hormones and glucocorticoid. But those things are very important, and getting this kind of many variables or many proxies per individual, per sample, is kind of the holy grail. both behaviorally and physiologically from the same individual would be amazing. Okay, and so then I'll jump into basically 3 papers from my PhD. These are poorly screenshotted titles from the journal, but yeah, you can find them like that, or on my ResearchGate page if you want to read more about them. So in my first paper, we basically wanted to do like the classic study where we, or build on the classic study where we wanted to combine both human disturbances and environmental disturbances and see how they would affect the glucocorticoid concentrations in wild impala. So again, the reason why we did this is because glucocorticoids are a proxy for the physiological stress response, and the stress response would show us how individuals or how animals are reacting to different kinds of non-lethal disturbances. And the effects that we hope that the effects that we saw in common antelope like the impala could also be transferable to rare antelopes or other animals in the same system. But mostly most of the studies that have been published or had been published on this kind of topic, they basically just used human disturbance and didn't really consider environmental disturbances. And when they did, they often didn't have any spatial or temporal variation in there. It was just like inside, outside the park, for example, or it would be wet and dry season, but there's no spatial variation or temporal variation in these disturbance proxies. So we wanted to like delve into that and see what the value of that is. So what we found was that a human disturbance does significantly increase glucocorticoid levels in impala, correcting for everything else. And as expected, forage quality does also, so increasing foraging quality decreases glucocorticoid levels. We hypothesized this because with higher NDVI, this means greener area, there's more high quality food available, and so everything would be easier to deal with. And so glucocorticoid levels should decrease. This has been shown in literature before. But when we delved into the results and into the discussion, we found that although the human effect was there, it was quite small. It did not explain a lot of variation, while the foraging quality was, maybe unsurprisingly, very important. And impala in areas or in times and areas where the dry season was at its harshest, they had by far the highest glucocorticoid levels. And so we kind of, this is not very nuanced, but we in the paper we kind of go into more nuance, and we say that we basically conclude that although humans might have a big effect on animals, on their physiological state, they might not have a massive effect, at least not compared to important environmental disturbances like forage quality. And so making sure that climate change or rivers, ephemeral rivers remain or have enough water throughout the wet season, those kind of things are very important. So basically land use changes might be very important or more important than keeping certain human disturbances at bay. So the second paper was focusing on a different hormone. So triiodothyronine, or thyroid hormones in general, are often used. Yes, are often used as a proxy for nutritional stress. So thyroid hormones, TH here, is related to metabolic rate and thermoregulation, but most studies are done experimentally. Very, very few studies have been done on wild populations. And those that have been done experimentally, they're basically focusing on the nutritional part of it, or the metabolic rate part of it. And often it's just they have control animals and experimental animals, and then just starve the experimental animals and see what it does to their physiology. So yeah, we expected that thyroid hormones would be mostly driven by NDVI or forage quality, and much less so by everything else. What we found was that there wasn't a significant effect of NDVI as we expected. However, temperature had a much bigger effect on thyroid hormone levels. So much so that it kind of negated the effect of NDVI or forage quality, And so we're thinking that in impala populations, thyroid hormones might be a better indicator of thermoregulation. And there's a whole body of literature on how thermoregulation is important in tropical animals, where you could think, it's warm every day, which it is, but there is variation in seasonally. And so we posited that thyroid hormones in wild populations might not only respond to nutritional state, or might only respond to nutritional states in extreme situations like starvation. But since we only selected individuals that had a good body condition, so that did not show any signs of starvation, we might not have picked up on this pattern. So for us, in our individuals, thermoregulation seemed to be the driving force of variation in thyroid hormones. And so then we were thinking that climate change and the variation in extreme temperatures that seems to become more common might be a bigger threat to animals or impala population. Okay, and then the last paper is something completely different. Here we looked at the social environment, basically, and their effect on glucocorticoids. So we were interested in how territoriality in impala affects their glucocorticoid levels in territorial males versus bachelor males. So bachelor males are those that are just in a group with other males, and they don't have a territory. They just kind of walk around. Yes, there you go. So we hypothesized that since territoriality is essential for reproductive success, but it is a very exhausting occupation, we expected that glucocorticoids would be much higher in territorial males compared to bachelors. That was kind of the baseline. However, we expected that when the environment or foraging quality was bad, or forage quality was bad or low in DVI, we expected then the stress would compound in territorial males, so they would have a much higher glucocorticoid levels compared to bachelors, because they would have to deal with the lack of food and having a territory. Secondly, we thought that with increasing population density, when there's more males around, their glucocorticoid levels would go up because of mate defense. And then lastly, if they had high or bigger groups or herds to protect from other males, their glucocorticoid levels should go up as well. This was an interesting paper because we found nothing like it. So the territorial males had much higher glucocorticoid levels than bachelors, but this was irrespective of forage quality. So there is a tendency for lower glucocorticoid levels with higher foraging quality, but this was not significant. While bachelors show this classic effect that we saw over the entire population, where with females, for example. We did not see an effect of population density, so both or glucocorticoid levels went up for both bachelors and territorial males, while territorial males will still, of course, higher glucocorticoid levels. And then lastly, herd size did not affect GC levels in territorial males. Obviously, bachelors don't have a herd size. And so what we found, or what we decided or concluded from this, was that even though glucocorticoid levels are high, it seems to be regardless of any disturbances. And so this might relate back to what I mentioned all the way in the beginning, where glucocorticoids shouldn't be seen as this negative repercussion of things, of stressors, but more of an adaptive mechanism. Like glucocorticoids are this adaptive mechanism that allows or facilitates territoriality, which is essential for reproduction, and therefore it ultimately increases overall fitness. Even though having this elevated glucocorticoid levels for a long time, like chronically, does deplete their energy reserves. And after 2-3 months, we see from observations, these males lose their territory because they're too weak, because they very rarely eat. They basically just herd and defend their herd of females, and then they mate as well. But so yeah. Okay. So in summary, I don't know what the time is. In summary, when we are measuring these disturbances to animals, we found it was quite important to explore spatially and temporally explicit proxies of disturbance, both of human disturbance and environmental disturbance, and to consider them at the same time, both at the same time, because otherwise we might focus our conclusions more on one or the other, especially towards human disturbances, while we don't account for a lot of the variation in, for example, stress physiology or behavioral stress response. And then this was not mentioned here in the results, but we found that it was very interesting to complement both physiological stress response to behavioral observations, because honestly, the stress response is twofold. It really is both behavior and physiology. And so one of the clearest examples of that would be if an animal is suffering from heat stress, it could gear up its thermoregulation and have a lot of physiological adaptations, or it could go into the shade and move to have a behavioral reaction and move into the shade. And so those two are quite important to understand, that physiological responses are often secondary to behavior, even though physiology can also modulate behavior. But that's a conversation for another time. And lastly, so hormones as a proxy of stress. Yes, we can use hormones as a proxy of stress, but the stress response is an adaptive mechanism. So hormones are generally beneficial, not just costly, although they can have apparent maladaptive consequences. They often result in an overall fitness benefit. And glucocorticoids shouldn't really be equated to stress hormones or stress. They could be considered even anti-stress hormones because they basically help us deal with stressors. So yeah, that was basically my presentation, or very short summary of my PhD, and I'll take any questions. Thank you.
Lui nous a présenté aujourd'hui donc l'adaptation au stress environnemental et les leçons retenues des antilopes africaines dans le cas de l'impala. Merci à lui donc de nous avoir présenté donc cette belle présentation qui nous change de l'ornithologie. Dans tous les cas, vous savez que les oiseaux cohabitent avec les mammifères dans les différents milieux. Donc, c'est tout à fait correct que de temps en temps, nous ayons à sortir un peu de notre cadre habituel. Comme le disait Elie au départ, donc aujourd'hui, c'est la 60e section donc, de Friday for Bird, depuis qu'on a commencé depuis 1 an à peu près et demi. Donc, lui nous a présenté ce test-là en commençant d'abord par nous définir ce que c'est que la perturbation. Il est revenu sur les questions de la physiologie des animaux comme un instrument pour la conservation, à travers donc la compréhension des mécanismes et de l'importance de la physiologie pour la conservation comme outil des conservateurs, donc des managers de la conservation, pour en faire revenir et nous dire que la physiologie de la conservation aide à la conservation tout court. Il indique également que le stress, tel qu'il le définit, n'est pas mauvais, puisque c'est un outil d'adaptation au milieu que les animaux ont. Donc, le stress ici est considéré comme un facilitateur plutôt que être considéré comme quelque chose de très négatif pour la survie des espèces dans les milieux naturels. En prenant donc l'exemple des impalas, il a démontré à travers ses travaux de thèse sur au moins 3 ou 4 articles que ce stress-là était plutôt un facilitateur. Il indique également que les impalas mâles sur lesquels il a travaillé, au niveau donc de ces zones-là, les mâles territoriaux émettent beaucoup plus d'hormones glucocorticoïdes que les célibataires, pour dire que les espèces considérées devait donc nous donner des éléments suffisants en vue donc d'une meilleure conservation des impalas. Il a longuement travaillé sur ces espèces dans la zone de Serengeti, qui est une zone importante pour la conservation de la biodiversité, ce qu'on appelle les hotspots de la biodiversité. Il est revenu sur plusieurs aspects comportementaux liés à la physiologie donc, des espèces qui nous permettent de comprendre bien l'importance donc d'étudier la physiologie des espèces dans un contexte de physiologie de la conservation afin de pouvoir donc donner des réponses adaptées à la conservation des espèces en comprenant leurs besoins physiologiques. Les éléments, comme vous le savez, seront donc encore une fois diffusés lors des différentes présentations. Donc, je vais laisser la parole à Ellie pour qu'on commence donc les séances d'échange. Merci à tous